In the quiet corridors of MIT, a team of chemists took aim at one of nature's oldest and most stubborn molecules, Rubisco. For decades, this enzyme has been both a marvel and a mystery. It jumpstarts photosynthesis, the process that powers all life on Earth, yet it's notoriously sluggish, a biological bottleneck. Now, using a breakthrough technique called continuous directed evolution, the MIT team has managed to re-engineer Rubisco, making it up to 25% more efficient. It's more than just a scientific upgrade. This lab-evolved enzyme could help crops grow faster, make plants more productive, and transform global agriculture as we know it. During photosynthesis, Rubisco performs a simple yet vital task, capturing carbon dioxide and turning it into life-sustaining sugars. But there's a catch. Rubisco, despite being the most abundant enzyme on Earth, is painfully slow, handling only a handful of reactions each second. Even worse, it often gets confused, reacting with oxygen instead of carbon dioxide, wasting precious sunlight in the process. To tackle this age-old inefficiency, MIT scientists turned to evolution itself, but sped it up. Using their Mutot-7 system, a method that lets evolution unfold inside living cells, they created thousands of enzyme variants and let nature decide which worked best. This is a compelling demonstration of how we can truly improve Rubisco's enzymatic power, said Professor Matthew Shoulders, who led the study alongside Robert Wilson and graduate student Julie MacDonald. Their journey began with a bacterial form of Rubisco, one found in a low-oxygen world where it already worked faster than most. They placed it in E. coli and exposed it to ordinary oxygen-rich air, forcing it to adapt or fail. Round after round, mutation after mutation, the enzyme slowly evolved, learning to prefer carbon dioxide over oxygen. After 6 evolutionary cycles, 3 powerful mutations emerged, tiny tweaks near the enzyme's active site that made a world of difference. Rubisco, once easily distracted by oxygen, now stayed focused on carbon. It's like we taught Rubisco to stop chasing the wrong partner, Shoulders explained with a smile. The result was a version of Rubisco that could work efficiently even in oxygen-rich environments, something nature had never quite perfected. And the implications are enormous. Plants lose up to 30% of their sunlight energy through photorespiration, a side effect of Rubisco's oxygen problem. Fixing that could mean higher crop yields, faster growth, and perhaps even a greener, more sustainable planet. This opens the door to a new era of enzyme engineering, said Wilson. It's a step beyond everything we've done before. Now, the MIT team is taking the next leap, applying this evolutionary technique to the Rubisco found in plants themselves. If successful, they might just rewrite the story of photosynthesis, and with it, the future of food and energy on Earth. MIT scientists have achieved the impossible, they've supercharged photosynthesis. Using a breakthrough technique called continuous directed evolution, they re-engineered Rubisco, the enzyme that powers plant life, making it 25% more efficient. This lab-evolved enzyme could help crops grow faster, boost food production, and transform agriculture worldwide.